Hello, I'm Martin Blaser. I'm the director of the Center for Advanced Biotechnology and Medicine, or CABM. Today, I have the pleasure of speaking with CABM resident faculty member, Dr. Bishr Omary. He is the Rutgers Biomedical and Health Sciences Senior Vice Chancellor for Academic Affairs and Research. He also is the Henry Rutgers Professor of Biomedical Sciences and a professor in the Department of Medicine at Robert Wood Johnson Medical School. Welcome, Bishr.
Thanks so much, Mari. Thanks for the opportunity. This is great.
Oh, thank you. So just to begin, I wonder if you can tell me a little about your education and your early career, the part of your career before you got to CABM.
Sure. So I went to undergraduate school at George Mason University, which is in the Northern Virginia area. I was a chemistry major. Then after that, I went to graduate school in chemistry department. But I was so lucky because I got a chance to work at the Salk Institute, which was very close to where UC San Diego is. That's where I was at. And in fact, my mentor there, I met him on a soccer field. and he was absolutely fantastic. So then I shifted more towards kind of cell biology and cancer biology. So that was graduate school. And then I did a 1-year postdoc. And it was really during graduate school that I got to learn about and appreciate. I was hardcore chemistry. Then I shifted more into biology of things. So I got interested in medicine, and that's kind of how I ended up afterwards going to medical school and then kind of staying in academic medicine.
Did you have to do the whole medical school, or did they give you credit for your PhD work?
Well, Mari, I was so lucky. I almost quit college because I was into sports when I played a lot of soccer when I was in college and worked in a restaurant. They wanted me to go into the management business of things. And I had a bunch of, my grades were either F's or A's. There was not too much in between. And then I decided that I'm just going to give it one more shot, one more semester where instead of going to the student union, playing foosball and ping pong and things like that. And for soccer games, the day before the game, you have to prepare for the game. So you don't go to any classes. The day of the game, of course, you've got the game. The day after the game, you're celebrating. So you don't go to classes either. It was kind of like that. But I was so lucky because then I just started going to the library more, studying, and that made a huge difference. And then graduate school after that. I went to a 2-year medical school. I was so lucky because at that time, University of Miami had a PhD to MD program. So that's what it was. So I ended up saving a lot of time this way.
Yeah. How old were you when you went to college?
I was actually 1 year. I was like 17 years old when I started. But then it took me 4.5 years to finish because I had a bunch of F's. And then I did some research in my last year into the year between 4.5 and 5, and so that's how it was.
I ask you that because I was 16 when I went to college, and I was just way too young. And basically, I didn't start studying till I was a junior, which is when I was 18. So I didn't really gain anything. Well, I had a good time, but I didn't gain that much from going to college so young. So great. And then what was the next stop after after UCSD? Then you were at University of Miami, and then what came next?
Yeah. So while I was because my GPA in undergraduate school was only 2.9. Because the first 2 years I barely kept, so I can just keep playing soccer. The last 2 years I did well. So when I applied to medical school, it took me actually 3 times to do this. My parents thought I was completely, crazy. And the program I really wanted to go to was University of Miami because I knew it was 2 years. I would save time. I applied to a bunch of schools. Then I was one of the youngest ones to get into because that program at the time, a lot of applicants, they used to get 800 applicants for 36 slots, but a lot of them were associate professors, basically, established faculty members who wanted to change the direction to go from being scientist into physician scientist. So that's how it was.
And then after medical school, then you did training in
yeah. So then I did internal medicine, then I did GI, and then started my first real job. That was at Stanford University as an assistant professor.
Yeah, and then gradually you worked your way up the ladder. I know you were the chief of GI at Stanford, and then you went to Michigan. You were chair of a basic science department, and had many really a terrific career, like as if you were, on the soccer field. So
Yeah, I was very lucky able to go from one place to the other. So, but I mean, I was only Stanford, Michigan. Now I'm so lucky to be here at Rutgers. Hopefully, I won't be moving anymore. So,
so let me ask you, how long have you been at Rutgers? How long have you been at CABM, and what brought you here?
well, what brought me here is an opportunity. I mean, Rutgers is as an institution, it's really an up and coming institution. I mean, they're a top 50 place, I would say. It depends whether you use NIH ranking criteria and so on. But I, um, I had a connection to find out about this position. It was really a sort of an accident. And then the more I found out about it, the more intrigued I was. And then I was lucky that the Chancellor invited me to interview. And then as you learn more and more about something, you either decide maybe it's not the right thing for you, or you really get excited about it. And for me, it was becoming more excited about it. Um, and I've been here now for about a little over 15 months. So I started in September of 2019.
Great. So now we've talked about your career. Let's talk about your scientific work. What's the main focus of your lab right now?
Right now, we're working on two major areas. One is intermediate filament proteins, and this is something that I've been working on for 30 years. That's why I started my research career as a faculty member. But in the last maybe 6 to 7 years, we started through the back door working on a group of rare diseases called porphyrias. So those are the two major areas of focus in the lab.
And is there a connection between the filament proteins and porphyria, or they're different?
The only connection is that the protein aggregation. So the mutations in intermediate filaments lead to protein aggregation. In porphyrias, the oxidative stress leads also to protein aggregation, and that's actually how we ended up working on porphyrias.
Mm-hmm. Yeah, of course, protein aggregation is a common and important theme these days in degenerative disease.
Absolutely.
So how are you approaching some of the scientific problems that you're dealing with?
So early on, we were really trying to understand in terms of intermediate filaments. No one really knew exactly what the function was. So a lot of the work that we did trying to figure out how they worked using post-translational modifications or identifying associated proteins as a handle to understand how they work, but also using genetic models, because maybe the disease if there's a disease, then so we used those kinds of approaches. And then, but in the last few years, there's more than 70 different human diseases that are caused by mutations in this family of proteins, and there's actually for none of them there's any treatment per se. So you treat the consequence of the mutation, whether it's metabolic disease, neuropathy, myopathy, things of that sort. So we started kind of a drug screening effort for that, and also for the porphyrias. So that's the exciting part that we're working on now, because we have several hits that work in genetic models, for example, or work in zebrafish models in the case of the porphyria. So now we're moving to mouse models for the porphyria. So that's what we're working on to try to. And the cool thing about this is that the drug actually, if it works in terms of improving the phenotype, but when you try to figure out how does the drug work, in fact, you learn about the pathogenesis and in fact some of the pathways that are involved.
Yeah, great. Well, that sounds quite exciting. So let me ask you a more general question, which is, in your lab, how do you interact with students and trainees, the people you're working with?
Well, I try to do it as often as possible. I try to make myself available as much as possible. So it's a usual one-on-one. It's usual group meetings. Talking over the phone, and I interact with them sometimes so much that one of them recently asked me to be the officiant on his wedding. I'm very lucky. I've always been, had fantastic coworkers in the lab, so it's been great.
That's a great honor. That's terrific. So now let me ask you a rather odd question, which is, imagine we were writing your obituary. And the question is, how would you like to be remembered for your science? What is it that comes to your mind?
I mean, usually it's really what others, of course, think about one's work, but hopefully they will feel that we made a contribution in terms of understanding the disease relevance of the keratins in digestive or simple glandular epithelia, in terms of how do they work and what their significance is. And that also opened up a number of different kind of paths, if you will, in the field. So I think if they will say that, I'll be a happy guy.
Great, great. Finally, I know that you have other roles at Rutgers and outside of Rutgers. Can you just tell us a little about your important roles in Rutgers and in the greater scientific and medical communities?
Well, I think for Rutgers, you pretty much introduced my role here at Rutgers. Outside Rutgers, I'm involved in one of the gastroenterology organizations. It's called American Gastroenterological Association. And I have the privilege to serve as president of this GI society this year. First, you're a vice president, president-elect, president, and then past president. So it's been a fantastic experience. The organization has really just wonderful staff, and I have really wonderful colleagues to work with. And trying to make a little bit of an impact along the way to promote science, to promote training. I mean, that's really the fun part of this position.
How big is the AGA? How many
16,000, about 16,000, a little over 16,000 members.
16.
6, yeah, 16.
Yeah, sure. That's a big, that's a lot of impact. Well, Bishr, thank you so much. I think we have a taste of you and your career and your lab, and I hope people will find it exciting. So
thanks so much, Mari, for offering these snapshots. I think it's really great, and it's been fantastic working at CABM, having you as our leader. Thank you so much.
Thank you.